Hello, dear students. I, Dr. Subhas, welcome to this session on important question part 1. Today, I will cover some of the most crucial topics that frequently appear in your examination. Let's begin. First, let us understand secondary metabolites. Secondary metabolites are organic compounds produced by plants that are not directly involved in primary function like growth, development, or reproduction. Instead, they play vital roles in a defense against herbivorous pathogen and attracting pollinators and helping the plant adapt for the environmental stress. The major classes include alkaloid, for example, morphine from the opium poppy seed, papaver somniferum, that is the biological source, glycoside such as digoxin from foxglove from the digitalis species, terpenoids like a menthol from mint plants and mentha species, and phenolics, for instance, uh, tannins found in plants like acacia. So these compounds are found in many important drugs that we can able to tell over here. Moving on to an important biosynthetic route, the amino acid biosynthetic pathway. This is the cyclic acid pathway, and so from that we can able to get that various nitrogen compound containing secondary metabolites, including alkalides and cyanogenic glycosides. So key precursor and alkalides, they are from tryptophan, so leads to indole alkylide such as reserpine, tyrosine precursor for isoquinoline alkylide like a morphine, ornithine give rise to tropine alkylide, for example atropine. So these are the, you have to remember this question frequently they are asking, that you have to be remember. The next one, case study, that camellia sinensis. So from the tea leaf we can able to get the caffeine, theophylline, catechin, and tannin related polyphenols also. So Camellia sinensis, that is the botanical source. This family is the Theaceae. Here alkaloid, that caffeine, theophylline. So these are the alkaloids are present over here. Polyphenol, so catechin and tannin, which are responsible for flavor and antioxidant activity. Volatile compounds and some amino acids and a minor quantity of vitamins also present over here. Next, organized drug derived from some of the specific organized plant parts. Example, clove. So dried flower buds of the Syzygium aromaticum used as a carminative, antiseptic, and dental analgesic. Cinnamon bark, Cinnamomum xylonicum, widely used as a flavoring agent and carminative. These are the excellent example of crude drugs for the specific morphological parts. Case study, that mentha. So it is a mint leaf. Another one, it is one of the chemical constant. So other chemical constant, menthol, menthone, menthol esters, volatile oil. So these are the phytochemicals are having the therapeutic uses. Carminative, which means relieve the flatulence; antispasmodic, which means they reduce the muscle spasm; flavoring agent in foods and pharmaceuticals; provide cooling sensation in topical topical formulations, and reduce the cough and cold preparation also. We can able to use. Menthol is one of the most widely used natural cooling agent. Qualitative analysis, chemical test for asafoetida. So identification test for asafoetida, it is an oleo gum resin with a strong characteristic odor. Important chemical tests such as sulfuric acid test, which produce red to reddish brown color; ferrichlorate test, which gives blue or green color due to the phenolic compound; odor test, strong persistent like a garlic odor due to the sulfur containing volatile oil. Next one, the structure of the reserpine. Here we have a reserpine. So these are the naturally occurring indole alkylide. So this molecular formula, that is C33H40N2O9. It has complex structure with an indole nucleus attached to an ester system. Reserpine was one of the first effective antihypertensive and antipsychotic agent. So these are derived from that Ravolfia serpentina. So this is one of the that important antihypertensive drug that you have to be remember. Let us briefly recall primary metabolic pathway. So primary metabolic pathway, glycolysis converts glucose to pyruvate producing ATP and NADH. Krebs cycle, citric acid cycle, oxidize acetyl CoA to release energy. Photosynthesis convert light energy into chemical energy forming glucose. Pentose phosphate pathway generates NADPH and 5 carbon sugars. Understanding the difference between the primary and secondary metabolism is a fundamental. Next one, that Taxol, or paclitaxel also we can able to call. It acts as a mitotic inhibitor and it is used as a chemotherapy for the ovarian cancer, breast cancer, lung cancer. So it is a complex diterpenoid structure, makes it a challenging but rewarding this topic. Anti-hyperlipidemic drug. One example they were given here, gugulostirone they are asking. So gugulostirone is one of the phytosteroid anti-hyperlipidemic activity. It is isolated from the oleogum resin of the Combiphora mugul family Burseraceae. Gugulostirone is widely studied for its cholesterol lowering effects and represents an important traditional resin drug. I hope these points help you prepare effectively for your examination. Revise the sources, constituent users, and test thoroughly. Thank you for your attention. Wish you all the very best. Thank you.